So we're going to start chapter 3, which is cells and tissues. Let's first start with a review of the basic parts of a cell. The inside part of the cell is where the cytoplasm is. It's all the organelles and the fluid within the cell. There's also the nucleus, which is the center part of the cell. That's where our DNA is located. This is our control center. And all of that is surrounded by a plasma membrane, also called a cell membrane. It's made of a phospholipid bilayer. We've talked about phospholipids before, that special type of lipid that we reviewed in chapter 2. So it's made of a phospholipid bilayer that is basically forming the boundary between the inside and the outside of the cell. Here you can see a nice diagram of an animal cell, and you look at this and you're probably like, what the heck is all of this? But believe it or not, you'll know what all of these things are by the end of this chapter. My mouse is pointing to that plasma membrane, that outer cell membrane. If we were to zoom into that structure, this is what you would see. You see those phospholipid bilayer, those two layers of phospholipids. This is a special membrane protein. Those are embedded within the bilayer. These allow larger molecules and ions to come into the cell, and we'll talk about that later. The large structure in the center of the cell, which is the nucleus. This is where all the DNA is. Inside of the nucleus is another structure called a nucleolus. The nucleus itself has holes and pores in it so substances can get in and out. Just outside the nucleus is all of these membrane structures called endoplasmic reticulum, also called ER. It's called rough ER because it has all of these little blue dots, which are ribosomes. Ribosomes are the little protein factories, and we'll talk about those later. This orange structure here is the Golgi. Sometimes it's called the Golgi Complex. Sometimes it's called the Golgi apparatus. It's differentiated from the endoplasmic reticulum because not only is it tubeways and passageways, but it also forms vesicles. These vesicles hold various substances that need to be transported into or around the cell. Here you see one of those vesicles fusing with the plasma membrane. This large structure here is the mitochondria. This is the energy factory for our cell. Here you can see a different type of endoplasmic reticulum. This is called smooth ER. It's called smooth because it does not have the ribosomes on it. The rough ER was right here with all those blue dots, those little ribosomes. Here you can see some of the free-floating ribosomes, those blue dots that are gathered throughout the cytoplasm. Also in the cytoplasm are peroxisomes. These contain hydrogen peroxide to help break down various structures inside the cell. The lysosome is here. This is our digestion factory for our cell. You can also see all of these microtubules and microfilaments giving our cell a three-dimensional shape. They kind of form an inner spiderweb network inside of our cell, making our cell three-dimensional. Not all cells have these structures, but some of them have these cilia, which are hair-like structures. Some cells have centrosomes or centrioles. These are special organelles that assist with cell division. Some cells will have a flagella, which is a long whip-like tail. Not all cells will have those outer membrane structures, though. Let's move on from this diagram and start talking about specifics of the organelles inside of a cell. Let's start with the outermost organelle, that plasma membrane. It's a flexible barrier, as we mentioned, made of a double layer of phospholipids. Interspersed amongst the phospholipids are cholesterol molecules, as well as glycolipids and proteins. There are special types of proteins that are involved in our plasma membrane. Integral proteins are embedded within the bilayer itself. Peripheral proteins are to either side of the membrane. And then glycoproteins are usually on the outside of the cell. These have special sugars attached to them. They help with cell-to-cell recognition. Here you can see some of the proteins I was mentioning. Integral proteins here embedded in the phospholipid bilayer. Here's another one. You can see special glycoproteins made of sugar molecules coming out on the outside of the cell. You can also see a special peripheral protein here on the inside of our cell membrane. The little kind of goldenrod structures inside our phospholipid bilayer are cholesterol molecules. they help to provide some rigidity for our phospholipid bilayer. Other functions of our plasma membrane, other than for it to be a barrier between the inside and outside of the cell, it is also going to transport proteins into and out of the cell. It causes the cell to be what we call selectively permeable. It's kind of like the bouncer for our cell, picking and choosing what can come in and leave the cell. There's external glycoproteins called antigens. These antigens allow cells to recognize one another or recognize invaders. This is how our immune system functions. There's also special receptor proteins as well as enzymes which help with intercellular signaling. This animation here goes through some specifics on membrane functions. The cytoplasm is the name for the area in between the cell membrane and the nucleus. So it's basically everything inside the cell except the nucleus. It's composed of the cytosol, which is what I call the cell juice. This is the water, dissolved ions, proteins, ATP, fatty acids, and any other molecules that are within the cell. Then there's the organelles. These are the mini organs. We listed some of these already. Some of them are membrane bound, like the endoplasmic reticulum and the Golgi. Some of them are not membrane bound. They form more of a framework, like the cytoskeleton. Ultimately, all of these organelles have specialized functions. Let's talk about the cytoskeleton. The cytoskeleton is that protein network of filaments and tubules. There are microfilaments, intermediate filaments, and microtubules that compose our cytoskeleton. Let's talk about these parts more specifically. The microfilaments are small protein strands that provide mechanical support, and they help generate movement for a cell. These also help to anchor proteins in the plasma membrane, and they provide support for any microvilli that may be on the cell surface. Intermediate filaments are larger than microfilaments, but smaller than the microtubules, which is why they're called intermediate filaments. These hold the organelles in place. They also help attach cells to one another. Then we have microtubules, which are long, hollow protein tubes. They help to determine the shape and the movement of the cell. These are the, quote, bones of a cell. They also make up the components of cilia and flagella, which are extracellular organelles. Here you can see a breakdown of these various components of the cytoskeleton, the microfilaments making up the microvilli in this case. Here you see the intermediate filaments holding organelles in place, giving the cell our three-dimensional shape. And then the microtubules as that hollow tube. These guys, for instance, the centrosome, they form the centrosome rather. They help with moving structures inside the cell. Other structures that are involved with microtubules include centrosomes, as I've mentioned, centrioles, cilia, as well as flagella. Centrosomes are usually located near the nucleus. They are a pair of centrioles. They contain special proteins called tubulins that will build microtubules in the non-dividing cells. And those tubulins, those microtubules that are secreted from the centrioles, help to separate the chromosomes during mitosis, which is the process of nuclear division. Centrioles themselves are the hollow cylinders. They're made of 9 sets of 3 microtubules. The specific part of the centrosome that forms the mitotic spindle during cell division. So the specific part of the centrosome that's forming the spindle is called a centriole. The cilia are formed from microtubules. These are the short hair-like projections of the plasma membrane. The job is not to move the cell, but rather to move things across the surface of the cell. They have coordinated movements with one another to help move substances across the surface of the cell or circulate the liquid environment around the cell. Flagella are made from microtubules as well. This is a long, whip-like structure. This one's going to help the cell move. The most notable cell, or really the only human cell that has flagella, is a sperm cell. And this is going to allow that sperm cell to swim to the oviduct to fertilize an egg. Ribosomes are other cellular structures inside the cell. They're made from RNA. They're also made from proteins that are made in the nucleolus of the cell's nucleus. They have a large and a small subunit, and that large and small subunit are then assembled within the cytosol. They're located both along the rough endoplasmic reticulum, within the mitochondria, and they can also be free-floating in the cytosol. Ultimately, their job is to make proteins for our cell. So this is our protein factory. Here you can see the large subunit and the small subunit coming together to make a functional ribosome. The endoplasmic reticulum is the large membrane system leaving from the outer nuclear membrane through the cytoplasm. The majority of the membranes inside of our cell is made of endoplasmic reticulum. It comes in two varieties, as I've mentioned, rough and smooth, and they're usually abbreviated RER and SER. Here you can see the different structures. Of course, I mentioned the rough ER having those ribosomes on the outside of it, whereas the smooth ER is called smooth because it does not have the ribosomes. They're all continuous with one another. The smooth leads into the rough, and that leads into the nucleus. Ultimately, the rough ER is going to synthesize glycoproteins as well as phospholipids and transfer them to the plasma membrane. The smooth ER is going to make fatty acids, steroids, estrogen, testosterone. It also acts as a detoxifier, so it helps remove potentially harmful substances. It can also release calcium ions to trigger muscle contractions in muscle cells. The rough endoplasmic reticulum leaving that nuclear envelope surrounded by those ribosomes, giving it its rough appearance. Proteins are made there. They're inserted into that rough ER membrane, and they can either go to the plasma membrane or to other organelle membranes. The proteins that are made there can then go through the rough ER and then can be secreted out of the cell. Our smooth ER, as I mentioned, extends from the rough ER, and it's in a series of microtubules. There's no ribosomes on its surface, which is why it's called smooth. This is where fatty acids and steroids are manufactured. As I mentioned, it helps to detoxify our cells and our body as a whole. It can also serve as a storage place for calcium ions, for muscle contractions and hormone actions. In the muscle cells and heart muscle cells, it's called sarcoplasmic reticulum because of its specialized function. We'll talk about those functions when we get to the muscular system. The Golgi complex, also called the Golgi apparatus, is that series of flattened membranes or flattened sacs. It's found in most cells. The majority of our transporting is going to happen here with the formation of these vesicles. These transfer vesicles will contain special proteins which kind of bud off from these cisternae, which are these flattened tubes. These vesicles kind of bud off or bloom off of this structure and float to either the cell membrane so it can be excised or leave the cell, or floats to various parts inside the cell. Lysosomes can actually be made from the Golgi apparatus. They're membrane-enclosed vesicles. These contain digestive enzymes. They help break down material that are brought in by the cell. They can fuse with other vesicles or other organelles to help digest them. They can also release any remnants back into the cytosol for those remnants to be recycled. Here you can see a close-up view of a lysosome, basically a membrane sac with lots of digestive enzymes inside of them. Notice some of the functions listed here. Other than digesting substances that come into the cell, they also help digest worn-out organelles. They can also digest the entire cell. They can rupture and help the cell commit self-suicide, so to speak, so the cell can die for the greater good of the organism. If the cell's old and things aren't working properly, the lysosome will rupture and break down the rest of the cell. Peroxisomes contain special enzymes called oxidases. These enzymes help remove hydrogen atoms from molecules like amino acids and fatty acids. They also help to detoxify alcohol in the liver. Hydrogen peroxide is a byproduct of special chemical reactions, so the catalase within the peroxisome breaks down that hydrogen peroxide into water and oxygen. Hydrogen peroxide itself, as it accumulates inside of our cells from metabolic processes, can be harmful. Proteasomes are tiny barrel-shaped organelles. They contain lots of proteasomes in the cytosol and nucleus of each cell, thousands, in fact, inside the cytosol and nucleus of every cell. Inside these proteasomes are proteases. Proteases are enzymes that break down proteins. They're breaking down faulty proteins, damaged proteins, proteins that aren't needed anymore. Those parts then are broken down and turned into other enzymes or other amino acids and can be recycled into other things for the cell. The mitochondria has a fluid-filled compartment called a matrix. It has a double membrane. It's kind of a kidney-shaped organelle. The inner folded membrane is called the cristae. Oxidative phosphorylation takes place here. Movement of electrons through the cristae help to make the ATP, which is needed for cellular energy. They also help to regulate intracellular calcium levels. Mitochondria themselves contain their own DNA and their own ribosomes. Here you see a picture of them. Again, a long kind of kidney-shaped or sausage-shaped organelle. You see the inner cristae, or those inner folds, where the oxidative phosphorylation will occur to make ATP. You can see some ribosomes gathered in there. There is also DNA in there, however, it's not pictured on this slide. The nucleus itself is comprised of the nuclear envelope, which is the membrane that encloses the nucleus. It has nuclear pores within it, which are the small holes allowing things to come into and out of the nucleus. The nucleolus is the large round structure that's made of DNA, RNA, and proteins. This is where ribosomes are made inside of our cell. In fact, it's where the large and small subunits are made. The large and small subunits then come together within the cytosol. The nucleus also contains chromatin. Chromatin is the loose, kind of unwound form of DNA that is inside of our cells. When cells are not dividing, when they're doing their job, when they're living their life, DNA is not found in chromosome form, it's found in chromatin form. Chromosomes are the condensed form of chromatin. If you can imagine taking a string and holding it in either hand and twisting both ends of that string, that DNA is going to condense over and over and over and twist over upon itself, forming chromosomes. However, the majority of the time the DNA is in chromatin, or nice relaxed form. Here you can see what I mean with the nuclear envelope being that outer structure. You can see a little bit of that rough ER in this picture. You can see those nuclear pores as well as the nucleolus inside of our nucleus. The extracellular fluids, these are all the fluids outside of the cell. It comes in different varieties, different names, depending on where it is in the body. Interstitial fluid is the name for the fluid that's in between cells. When we use the word plasma, we're talking about the fluid around blood cells that are within a blood vessel. When we use the term lymph, this is the extracellular fluid within a lymphatic vessel. When we use the term cerebrospinal fluid, this is the special fluid that surrounds the brain and spinal cord. All of these fluids are, in fact, interstitial fluids. They're specialized, and they're all outside of the cell. We're going to stop here for part 1.